Okay, this is Bio 181, photosynthesis, and this is where we're going to get into more detail in terms of the light-dependent reactions and the light-independent reactions. So the first part is the light-dependent reactions. The light is going to come in from the sun. Okay? It's going to take place within, so I'm going to go over here up at the top. So this is the chloroplast. In the chloroplast, you've got these thylakoid membranes. Okay. So this is the thylakoid, Ch8. Okay. So, and again, this whole thing is the chloroplast. Chloroplast. All right. So I'm now going to just zoom into the thylakoid membrane. So I'm taking the single one here. That's what I'm taking. So this is often where I lose people. So here's the single membrane. I'm within the chloroplast. That's what I'm looking at now. Okay. So here we go. So I'm taking the thylakoid here, right? And this is a stack. So there'd be another one below it. This is a phospholipid bilayer. Okay. This is the thylakoid, Thylakoid, right? Within the chloroplast. There are many of these. All right. So what happens is light energy is going to split water. So we'll walk through this. So water is one of the reactants in photosynthesis. So you've got water in here, okay? And the light energy is going to be absorbed in a photosystem. So you've got a small cluster of molecules in the photosystem in the membrane here. So in the thylakoid membrane, you've got a photosystem. In that photosystem, the light is absorbed. The electrons are going to be excited in the molecules, in pigment molecules like chlorophyll. So this contains lots of chlorophyll, which is the name of a pigment molecule that absorbs light, okay, along with carotenoids. So the electrons there are very excited, okay? They travel to a reaction center. They get amplified, so the energy is increased, and that energy is used to split water. So water is split into oxygen gas, which then diffuses out of the cell, out of the thylakoid, out of the chloroplast, out of the cell. It also splits into hydrogen ions. So I'm just gonna, right? The hydrogen in the water is now going to be split into hydrogen ions. So it's plus, that means the electrons have also been removed. So the electrons have also been taken off. So there are electrons. So the energy from the sun is now, if we were to find it, it is now in this electron here, right? If we follow the energy. So from the sun, concentrated in the photosystem, transferred to the electron. Now that electron is going to be passed to a series of molecules, proteins that are embedded in this thylakoid membrane. And this is the electron transport system, the electron transport system. So it consists of these proteins here. It's going to transport this electron through it. The purpose is, as the electron goes through, some of the energy is going to be taken out of this electron, and the energy as it's taken out of the electron is going to be used to pump hydrogen ions into this space. So we're creating a little bit of a battery, and it's going to be a proton, right? Gradient. So we're creating gradient, a whole bunch of protons, which are all positively charged in the same area. They don't want to be together. They want to get apart, right? So we're creating this ion gradient or a battery, a whole bunch of protons, a whole bunch of hydrogen ions in this space. Then the electron, when it's done, it's actually going to get passed to an electron carrier. So the electron carrier without the electron is called NADP+. And then once it gets the electron added to it, actually two of them, once it gets two of them, it'll be called NADPH. Okay, so this form here is the form that has the high energy, so it's reduced. Whereas this other version is oxidized. Okay, so these are going to go to the next set of reactions, and they're going to bring the high energy electrons with them. The next thing that happens is with the hydrogen ion gradient, the exact same thing happens that occurs in cellular respiration is you have ATP synthase, which acts like a little rotary motor. It's stuck in the membrane, and the hydrogen ions are going to be allowed out, but the only way that they are going to be allowed out is if they make ATP. So the process of moving through this physical enzyme complex is going to rotate this part of it. It's going to push a phosphate onto ADP, adenosine diphosphate, and it's going to make ATP. And then that ATP is also going to be taken to the next set of reactions. Okay. So again, this process in here is called chemiosmosis. That is the process of driving the hydrogen ions into this enclosed space across a membrane. And then here, when we take ADP and make ATP, we are using ATP synthase. Okay, so again, this molecule here is ATP synthase. It is an enzyme complex. Okay. The second part, so we're coming in with ATP. So we made the ATP in the first part. We also made the electron carriers in the first part, in the light dependent reactions. Okay? So we're coming in with high energy electrons, and we're coming in with energy that we can use to make sugar, right? So we're coming in with electrons. We're actually coming in with some protons too that are in NADPH, 2 electrons and a proton. Anyway, carbon dioxide, the main idea here is that we're fixing carbon dioxide. So this is carbon fixation. Okay? It's also called the light independent reactions. And carbon fixation means that we're taking carbon dioxide from the gas form that's found in the atmosphere, and we're actually going to take that, and ultimately we're going to make glucose with it. So we're going to way down here make a product called glucose. That's the purpose of photosynthesis. We're going to make C6H12O6 ultimately, right? So carbon fixation is that process. So how do we do that? How do we get from carbon dioxide to glucose? That's what we're going to talk about now. I'm going to erase this a little bit here. That's where we're ending up. There are 3 stages that you want to know in carbon, in the Calvin cycle. So this is a cycle. This process is represented by the Calvin cycle. And it just repeats over and over again. So we're coming in here. I'm going to start with CO2 at the top. And this carbon dioxide, again, this is a gas, and it's going to be combined with a molecule called RUBP. So we have some acronyms that you're going to just have to memorize. You don't want to memorize the big word, which is ribulose bisphosphate. And so let's just combine these. This molecule here, this is a carbon molecule. It's a 5-carbon molecule. It's going to combine with this molecule, which has 1 carbon in it. It's going to make, really briefly, a 6-carbon molecule that is then going to split apart into a 3 carbon molecule. So you're going to have 2 3-carbon molecules that are made pretty quickly from the process of carbon fixation. So this process here of combining RUBP with CO2 is called carbon fixation. So that's the first step in the Calvin cycle that you need to know. This is carbon fixation. We're taking carbon from what's called an inorganic form. I know it has carbon in it, but it's not biological. So here it's inorganic. We're combining it and we're putting it into a molecule that is ultimately going to be turned into sugar. The product here is PGA, and that molecule is the end of this first stage. So carbon fixation. Then we're going to take PGA and we are going to add a whole bunch of energy and electrons to it. So we're going to bring in electrons from NADPH. It's bringing in electrons. We're going to add those. Once it delivers the electrons, it turns back into NADP+, which will go and get more electrons and bring them back for the next cycle. ATP, which again, these were brought in from the first part of the reaction, the light-dependent reactions. ATP is going to drop off energy. So ATP is going to drop off a lot of energy, and it's going to end up producing ADP, right, and PI, and then those will be regenerated ultimately to make more ATP. So we're bringing in energy, we're bringing in electrons. This is how we are reducing the carbon molecule. So this stage is called reduction. So number 2 is reduction. Remember, reduction is gain. So oxidation and reduction involves gain. So you're gaining electrons, you're gaining energy. This molecule PGA ultimately turns into another molecule that's called G3P. And that molecule, when it leaves the cycle, will make glucose. So it'll turn into glucose in a later step. Okay? We're not going to go into that. So that's reduction. We're bringing in the energy, right? Add electrons. Just gonna write it out. Add energy. So the light energy was transferred to these electrons, which is now transferred into this molecule, which is going to make glucose. The light energy also was trapped and put into this ATP molecule, which is going into this molecule, which is going to make glucose. So that's how we're getting the chemical energy in glucose from light. Now, the last part is just regeneration. So some of these molecules will kick out and go make glucose. Other of them are going to go back and they're going to keep the cycle going. So this stage is just called regeneration. So number 3 is regeneration. And we are just regenerating RUBP. Okay, so we're going back here. RUBP, we're making it again. It does take a little more energy, so we're going to add in a little bit more ATP during this process. Okay, again, that ATP here. So the ATP is from the light reaction as well as the electron carriers, and that's what's ultimately going to make this organic molecule down here, this carbon molecule, which is then going to make glucose. All right. Okay, the end. Oh no, this is the summary slide. I won't go over the whole thing again. This is from, I believe this one is from out of your book, right? So the sunlight's coming in. This is the photosystem where the light is absorbed. It's not showing all of the electron transport chain, but what will happen is that the hydrogen ions are going to be placed in here. They're going to fill up this internal space. This internal space is then going to be filled with ions that want to get away from each other, they are going to be allowed to get back out if they go through ATP synthase. Through that process, the hydrogen ions will leave and they form ATP. The electrons form NADPH. Both of those go into creating the next part. Again, this down here, the Calvin cycle. This is the light independent reactions. This is where carbon dioxide comes in, and ultimately we get G3P out, which is going to be used to make glucose. Okay. All right, that's the end. Oops. Oh, I know I said that. So the key concepts, you have these light energies used to split water molecules. You can read through the ones in the study guide for the key concepts. They're basically explaining succinctly the important parts of photosynthesis.